Hi everyone, welcome to your pre-lab for your photosynthesis disc lab. I'm going to be going over the procedure with you guys as well as establishing what you will be, the data you need for your control. Each group will be given a different question, and this is what you're going to be investigating in the lab. You'll also be given a rubric for the poster that you will turn in as the test grade. At the end of this video, we are going to put a list of all the materials that are available to the class in general. Know that you may not need all those materials. It just depends on what specific question you are investigating in this experiment. So the goal of this experiment is to record the number of chads that are floating in your carbon dioxide solution. So chads are just these little discs made from living plant tissue, and inside the discs are pockets of air that were produced through photosynthesis. So what we're going to be doing is we're going to first remove any additional air or any air that might already be in the chad. We're going to shine light on them to encourage the light-dependent reactions of photosynthesis to take place, and recording how many chads are floating at each minute interval during our investigation. So what I'm going to do is I'm going to show you how we're going to remove the air, and then we will switch camera angles to show you from the side the chads as they are floating, as they are performing photosynthesis, how many are going to be, or how many are floating at each of the time intervals. Now the chads themselves are just cut from living pothos ivy. Each chad is 0.5 centimeters across in diameter, and the light source we're going to be using is 20 centimeters from our sodium bicarbonate solution, which is going to be the source of CO2. We prepared this solution by mixing 1 gram of sodium bicarbonate in 100 milliliters of water, and now what I'm going to do is I'm going to remove the air from inside the chads themselves. And to do that, we need to create a vacuum. So we're going to create a vacuum using just a syringe and removing the plunger and putting the chads carefully into the barrel of our syringe. When we add the plunger on top, we're going to be using 10 chads, but when we add the plunger, we are going to want to be careful not to smash any of the chads. We're not going to go all the way to the top, just enough to get us started. And we're going to use this special solution to suspend and remove the air from the syringe itself. So the way to do that is you'll notice that when you put your chads into the syringe, they do float. It's a little hard to see here because of the angle, but they do float. So to remove the air, what I'm going to do is I'm going to bring the plunger and my water line up to the top as much as possible. I'm going to put my thumb over the edge of the syringe, and I'm going to pull the plunger back, give this a little shake, and what this does is that it changes the volume inside the plunger, but doesn't actually change the amount of air or the liquid. And so that creates a vacuum, which is what we need to get these chads to sink. They may require a couple of tappings, and this process may require a few tries before you can get all the air out from those pockets. But remember, we want to measure the amount of oxygen that's produced in our environmental conditions. So I think one more time should be the trick. While you guys are doing this, I am going to tell you that the temperature of the sodium bicarbonate solution is 19.8 degrees. So we will have thermometers as well as rulers, beakers, cups, forceps, some basic supplies for you guys. All right, so you can ensure that you can make the way that you know that your chads have been removed of all their oxygen is that when you'll look through the side of the syringe, they have sunk to the bottom. Now, what I'm going to do is I'm going to place my chads into my beaker, and in the meantime, I am going to keep this covered to prevent light from coming in. But what I'm going to do is I'm going to put my chads into the beaker here, and I'm going to keep track of how many chads are floating at different different points. So I'm going to use minutes at different time points. And for each of these, I'm just going to record the number of chads that are floating. After I've done this, I'm going to then take this data, however long it takes. I'm going to take that data and I'm going to graph it. I'm going to do a best fit line to find what is known as the ET50. Now your ET50 is the number of chads. Sorry, let's try that again. The number of chads on your y-axis, and then each minute is going to be your dependent variable here. And then what you want is to use your data. You're going to do a scatter plot, find your line of best fit, and then find out the time at which 50% of your chads are floating. And then we're going to compare the time on the control to the time that you got to see if there's any difference. So I'm going to cut this video and switch it over to the next one so that you guys can see the chads actually floating.